Hi folks, welcome back. In this video I want to talk about how DNA is divided up into chromosomes. And to remind you of how this works, I want you to take a look at this slide again. So if you look at this slide again, we see how DNA goes from being a double helix molecule, which is then wrapped around the histones as beads on a string, which then gets twisted into the rope-like 30 nanometer structure, which then gets supercoiled into the 300 nanometer structure, which then gets wrapped into the 700 nanometer structure, which we can see as the chromosomes. And these chromosome structures again display the characteristic aspects of being designed. So let me address this. When the DNA gets divided into chromosomes, you must know where to divide it without destroying any genes or any essential sequences that are contained. There must be order and logic to how it's divided. Therefore, you must know the DNA structure and the genetic code. Now, I'm going to explain the genetic code more when I discuss the functions of DNA. But for now, let me just say that the genetic code is how the language of the order of the DNA bases gets translated into the language of proteins by deciphering the amino acids called for by the specific DNA sequences. So it's the Rosetta Stone, which tells you which amino acids are coded for by the sequences of the DNA bases, which are transcribed into RNA bases. And the key to this Rosetta Stone is that all of this is done in triplets. So it's done 3 bases at a time. And these 3 base sets of sequences are called codons. So the various codons have an associated amino acid that they code for. And like I said, I'll have more to say about this when we talk about these functions of DNA. But back to what I was saying. You must know the structure of the DNA and the genetic code ahead of time in order to plan to strategically, intentionally, and intelligently create the structure of DNA and its genes with a design that allows you to divide it up into chromosomes and yet remain intact. Without knowing this ahead of time, you'd never be able to house the DNA as chromosomes inside the cell nucleus, and therefore DNA would not be the essential part of the structure of life. But not only that, you must also account for the structures of all the ends of the DNA. And for that, you must understand telomeres. Telomeres would be found here on the ends, as this arrow is pointing out. So we're talking about the ends, right? On those ends, you're going to find telomeres. They're the DNA regions at the ends of the eukaryotic chromosomes that contain sequences of direct tandem repeats. For an example of what that would be in humans, it would be the DNA sequence of TTA, GGG, that's repeated over and over. These repeat sequences enable the DNA ends to adopt a quadruplex loop conformation, which ends up looking something like a star. And that ends up on the ends of the chromosomes. These telomere caps protect the ends of the DNA, and they prevent the DNA repair mechanism from treating that DNA like it needs to be repaired. Now, telomeres can also form structures called T-loops and D-loops through interactions with specialized proteins. But these telomere structures are essential for the stability and survival of the chromosomes. They keep the ends intact and keep them safe from being degraded. They keep the ends of the chromosomes from looking like a frayed piece of string that needs to be fixed, which would include removing some of it, like cutting off the frayed end of a piece of string. Without them, the ends of our chromosomes would get chewed up by our DNA repair mechanism. Telomeres are also essential for the proper replication of DNA. During replication of the ends of the chromosomes, a short stretch of DNA, where the primer was, would be left exposed and susceptible to degradation. This would cause the ends of the chromosomes to be shortened after each round of replication. However, this doesn't happen because of the telomeres. They ensure replication of the ends of the chromosomes occurs without shortening or damage to the ends of the DNA using a specialized enzyme called telomerase. They keep the ends intact during DNA replication. So this structure ends up being an indication of the age and health of a cell. Without them, cell death will eventually occur. And now I need to explain another unique and essential feature of the chromosomes and DNA, the centromeres. The centromeres are found here where I'm pointing to with this red arrow, so right in there. They're where each new copy of the chromosome, called the sister chromatids, are attached to one another. The sister chromatids are each half of this structure, which are copies of the chromosomes that have been made from the original. The sister chromatids attaching at the centromere is what gives the chromosomes that characteristic X shape. Let me draw it on there for you. You see that? How we got kind of like that and that attached there at the centromere? Now, let me further explain how this works. Centromeres are necessary for the proper segregation of the chromosomes during mitosis, the process where a cell divides into two daughter cells. They're also essential for the proper segregation of the chromosomes during meiosis, the cell division process used to create the gamete cells that have only one copy of our genome and chromosomes. Meiosis is cell division where the diploid germline cells, and diploid means they have two copies of our genome and chromosomes, they undergo not only mitotic cell division, but they have an extra division of the chromosome copies to where the daughter cells are left with only one copy of each chromosome. These cells are now called haploid, which means they only have one copy of our genome and chromosomes. This process is what forms the egg and sperm. Once duplicated for mitosis or meiosis, each duplicate copy of the chromosomes, which I said is called a chromatid, remains attached to its sister chromatid, the other copy, via a protein called a kinetochore. Each chromatid has its own kinetochore, which is attached to a region of the DNA called CEN, C-E-N, DNA. So the sister chromatids have proteins called kinetochores bound to each one of them, and these kinetochores are also bound to the CEN DNA sequence of the centromere. So all that explanation to say, during either mitosis or meiosis, two features that are critical for the stability of the chromosomes take center stage: the telomeres and the centromeres. The telomeres hold the ends together and enable them to replicate properly while keeping them from being degraded, while the centromeres enable the sister chromatids during mitosis or meiosis to segregate properly into daughter cells while preventing the DNA from being pulled apart. Without the telomeres and the centromeres, cell division, both mitosis and meiosis, does not happen properly or at all, and life cannot be passed on. Therefore, you must know the need for these features of the DNA. They're essential sequences and structures. The sequences required to interact with their respective proteins, like the kinetochore. The domains and features of the interacting proteins that enable it to bind the DNA at their required regions or sequences. The roles of all of these in cell division, both mitosis and meiosis, and the overall processes of cell division, both mitosis and meiosis. And boy, that's a mouthful. But you need to know all of that ahead of time. You must know all this ahead of time in order to plan to strategically, intentionally, and intelligently create the structures of DNA, its specialized regions and structures necessary and essential for cell division, both mitosis and meiosis, and the interacting proteins exactly as required in these essential functions of DNA. But these are not the only aspects of cell division that are important. The entire cell cycle is an extremely well choreographed procession that is littered with critical proteins functioning as signals, structures, scaffolds, etc. The cell cycle is what governs how long a cell goes about doing what it was designed to do, whatever that is, until it's time for it to divide and make two daughter cells from the original. The entire cell cycle, which includes phases called G0, G1, S, G2, and then all the phases of mitosis, and then C. All of that is dependent on a vast array of these proteins, such as things called cyclins, cyclin-dependent protein kinase proteins, the CDKs, cell division cycle proteins, the CDCs, E3, SKPs, colon, F-box complexes, the SCFs, anaphase promoting complex, the APC, CDK inhibitors, etc., etc. Now, all the names of these proteins is not what's important here. What's important to understand is that there's a lot of them doing a lot of choreography, each one critically important for the entire cell cycle to function properly. The cell cycle, cell division, and therefore life would not be possible without them. So on top of the design element we have here, once again, this is yet another massive example of irreducible complexity. And I'm actually going to talk about that further in some future videos. All these proteins must be present and functioning together at the same time, or none of the aspects of the cell cycle and cell division can happen, and therefore life can't happen. So they cannot arrive gradually, randomly, or independently. They must all show up together at the same time. Furthermore, it's not just the proteins themselves that are important. It's the signals that choreograph and coordinate the proper sequential order of their functions. And where do these proteins and the information needed to coordinate them come from? Where does all that come from? From DNA, of course. Each step and phase of the cell cycle, their proper ordering, each protein and signal is ultimately controlled by the information that comes from DNA. Therefore, you must know the need for each phase of the cell cycle, what happens in each phase of the cell cycle, their proper ordering, the proteins required within each phase, the proteins required to progress from one phase to the next, the genes required to create each of these proteins, and the DNA to accomplish it all of that. You must know that ahead of time in order to plan to strategically, intelligently, and intentionally create all these features exactly as required in the structure and essential functions of DNA. And all this, as well as the telomeres and the centromeres, does not come from randomness. Once again, randomness, which is the basis of evolution, does not and could not create such irreducible complexity and choreography in the cell cycle and cell division. Randomness, by definition, is the opposite of organization, logic, intelligence, order, choreography, and patterns. Randomness does not result from intelligence and prescience because it's random. If the randomness of evolution created all the signals and proteins required for every aspect of the cell cycle and cell division, well, let me stop right there, because it couldn't. Randomness would have created a complete disarray of all of this, and it would be such a mess that all these processes and cycles would never even be able to get started. And the fallout from all this would be that their functions would be lost and life would not be possible. Therefore, randomness, which is the basis of evolution, would not and could not create all these life-essential aspects of the cell cycle and cell division, because randomness does not result from intelligence and prescience. And intelligence and prescience are absolute requirements for the creation of the cell cycle and cell division. And folks, I think that's a good place to end this video. So that's what I'm going to do. But come on back for the next one. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching. And as always, we'll leave the light on for you.